In this video, we will discuss about one of the examples of the interaction between glycans and cell adhesion molecules, and we will also discuss how these glycans will change in glycans will impact the cell adhesion molecules, and then it might further leads to the metastasis of the cells, specifically the tumor cells. So these glycans that are usually found on the glycoproteins that are present on the cell surface membranes, and these glycans helps in the interaction or adhesion with its neighboring molecules. And some of these glycan adhesion molecules are known as these IgCAMs, such as ICAMs and VCAMs1. uh, and this CAM means this cell adhesion molecules is the short form of this. uh, CAM is the short form of the cell adhesion molecules. So what happens because of the tumor microenvironment that is generated across the tumors, and it might leads to alter the glycosylation through various pathways, through various ways, such as this the cytokines that are present in the tumor microenvironment, the hypoxia. That tumor microenvironment create a condition uh, that is less amount of oxygen, that will cause the hypoxia, and the metabolic changes also occur, so that this tumor will continue to acquire their uncontrolled growth rate, and at the same time it will reduce the energy supply to the unnecessary things uh, from the tumor's point of view. So these are the major things that might cause alter in the glycosylation, and this altered glycosylation mainly occur in the ER and Golgi, or endoplasmic reticulum and Golgi apparatus. And this altered glycosylation might cause alteration. The altered glycosylation might impact the glycan profiles on the cell surface membrane, that is shown by here. That is this star shape interact the glycans that are uh, present on the uh, cell surface membranes glycoprotein. And this altered glycosylation might impact the lectin binding affinity, and this lectin binding affinity might leads to the weakening of this cell adhesion to its neighboring molecules, and that will leads to the following things, such as this proliferation, migration, survival, and permeability. These things might get affected because of the altered glycosylation, as the interaction between different cells because of this cell adhesion molecules glycan profiles got disturbed. And this not just the cell-cell interaction, the cells also for their adhesion property also acquired by the cells interaction with the extracellular matrix, and that will also get affected by the altered glycosylation of the proteins, that plays an important role in uh, adhesion, that plays an important role in extracellular matrix with extracellular matrix interaction. Then the lectin binding will also get affected because of the uh, altered glycosylation in this RTKs, that is receptor tyrosine kinase, such as BGFR2 and FGFR1. This will also affect this lectin binding affinity. So that will also contributes towards these factors, that is proliferation, migration, survival, and permeability. Now let us go through the impact of this altered glycosylation that might even lead to the metastasis. So consider this blue circle as tumor cells with altered glycosylation. This altered glycosylation is marked by this dark blue or purple glycan structures, and that will have the different glycans. And one of the glycans that is mentioned here is galactine 1. And because of the altered glycosylation, its cell adhesion properties get disturbed, and they will start to move, and this intravasation of these tumor molecules into the blood vessels occurs. And then because of the endothelial glycans such as E-selectin and galactine 3, that helps in this rolling adhesion and this extravasation of this uh, metastatic tumor into the secondary organ, that can be seen here. Here, this galactine 3 and E-selectins, they interact with the altered glycan profiles on the tumor cells, and helps in the rolling, decreasing the helps in this rolling and adhesion, that results in decreasing the speed of these tumor cells. And thereafter, these glycans also uh, supports or also complements the extravasation process, and thereby this tumor cells will be able to metastasize at the different metastasize at the different or secondary organ. And here this open circle represents the glycans that does not favor adhesion. So at the site of tumor infection, uh, the glycans on the tumor cells and glycans on the endothelial cells uh, will not favor adhesion, whereas the glycans on the extravasations or on the secondary organ, the they favors the adhesion of the aberrant glycosylation that occurred in the metastatic tumors. So these things promote the metastasis of the cells from one part of the from the site of its initiation origin to the secondary organ. I hope you have understood how this glycans plays an important role in cell adhesion, and that might even contributes to the metastasis. So, if you need to understand more about this glycan interaction and its role on cell adhesion and metastasis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.